Grazie. 15 minutes strict, please.
Okay, thanks very much. Thanks for the invitation. Can I have the first slide? La prima diapositiva, per favore. Okay. Thanks very much. Thanks for the invitation. It's a pleasure to present today the legacy, the Italian legacy for the visual imaging of the brain. And we have to go back to the 1700s, where a great anatomist like Gennari Santorini that studied in Pisa, and you'll see Pisa will be an important center for visual science. He, for the first time, perhaps, is the first demonstration that I know that really show that experience can change, can mold your brain. Indeed, he observed something that now Pietro is an expert of it, Pietro and Emiliano. He observed that acoustic nerve is much more prominent in people that are blind. And this is really something that still we don't understand the mechanism. Perhaps it's the myelin, we don't know. But perhaps the most famous Italian people that everybody knows is Gennari, another great anatomist that indeed we call the primary visual cortex triad cortex, because Gennari discovered this incredible, it's very hard for me to point, but doesn't matter, this incredible stria that is the input of the optic radiation from the thalamus, lateral genicular nucleus, to the cortex in layer 4. And now we know a lot of the organization of layer 4. We know that this layer 4 receives independent input from the left and right eye, and this input goes and builds what is called now ocular dominance, mapping of ocular dominance column. Now, where we know a lot, and a lot to the contribution to our knowledge about the organization, microscopic organization of the neural pattern in V1, are derived from the vision laboratory in Pisa, led and started by Lamberto Maffei and Adriana Fiorentini in the 70s. And I think that the greater contribution of this lab was also to link architecture, neural firing with function, how we see. And that is really what most of us does with functional imaging, really the link between function and local organization. And we know also, going back to Santorini, that this ocular dominance column can change during the critical period, and indeed experience mold our brain. But we can change dramatically this territory, this cortical territory dedicated to eye, mainly during the critical period. And later on in life, we think that we cannot change. So this has been a great dogma for many, many years. However, it will be important to reopen the critical period later in life, because we can in this way promote self-repair of the brain, and we do need to learn how to do so. So I think one of the last contributions of Lamberto, very important, was really to show that he can reopen critical period in the rat by pharmacological mean, but is a rat. What about human? And again, another great contribution comes from Adriana. We just lost recently Adriana, which she showed, perhaps it was the first time, that the first demonstration that indeed the human brain late in life too can, the primary visual cortex, can be reorganized plastically through perceptual learning. Yeah, but what about ocular dominance? This is perceptual learning, it's very local circuitry. Can we also change ocular dominance in adults? And here I move to what we are doing now in the Pisa Vision Lab. And this was a question that we asked a few years ago, but instead then go and measure and track what has been always considered Hebbian plasticity, the fact that if you don't use for a long time one input, the cortex will suppress that input, we went and studied the other side of the coin that we call homeostatic plasticity, that is really related to the fact that after short-term deprivation, the brain high up is getting the gain up of the deprived input. So you patch one eye, and what is happening? Well, you first measure ocular dominance perceptually, and usually it's the vision between the two eyes balanced. Then you patch one eye for 2 hours and a half, and what is happening? That input, that vision becomes much stronger. Is prevailing over the vision of the other eye by a factor of 2 or more. Well, but what this does means, do we have exactly the same mechanism that we have during development? So we studied the mechanism underlying this phenomenon using ultra-high resolution field that we need to do because we had only 10 minutes because the effect is not permanent, it's decaying very fast. And we use both we measured GABA, that is one of the implicated molecules during the development, the one that seems helping closing the critical period during development, in a scanner with Claudia, Emir, and Olli in Oxford, and more recent in a bold experiment in Pisa with Michela, Paola, Jan, and Laura. So let's see first the GABAergic or the data on GABA. So we measure GABA before deprivation, after 2.5 deprivation, in a voxel that was positioned on the occipital cortex, and we found the result only on those voxels, not in any other part of the brain, like parietal cortex. And you can see this is a GABA of one subject before deprivation. This is GABA measured in that voxel after deprivation. And you see that is decreased. Well, it's one subject. Well, we repeated in 20 subjects, and you see there is a clear decrease of GABA after 2.5 deprivation in the resting GABA, so with eyes closed. But what we think is more important, because it's a linking function to imaging, is that the people that had, in the subject in which we measured the strong decrease of GABA, are also the subject that showed the stronger effect of homeostatic plasticity, the one in which the boost of the deprived eye was stronger. That is GABA, let's see the bold, is also this boost associated with a local increase of activity. So this is the data, the experiment that we just published, that we presented in these 10 minutes that we had before deprivation, a series, sorry, it's not going, yes, a series of stimuli, low and high spatial frequency. Here I'm reporting the mapping of the response in along V1, V2, V3, and V4. This is the stimulation of the deprived eye before deprivation, and this is after deprivation. Notice by eye that here the response is larger. If we go and see the non-deprived eye, the response is decreasing. So the eye that has prevailing vision has also higher bold activity, particularly strong in V1. So V1 changes. So when we go and do all the analysis and ANOVA, we found that the effect is highly significant, but again, what is again important, people that did not have a strong functional effect also did not have this boost of the high spatial frequency in V1, and the correlation is quite good. So this correlation really suggests that there is a strong link between GABA and bold manipulation by change after deprivation with the function. But this is V1. If you are interested to this story, go and please and see the poster that Paola will present tomorrow, where we see this effect even in LGN. And this is again is another large, big dogma that says rely plasticity just at cortical circuitry. Okay, but you should tell me, well, why you are so much interested in this short-term transient plasticity. I mean, who cares? You cannot really change the brain in 10 minutes, well, perhaps we have means, both pharmacological but also more physiological means, in which we can stabilize that plasticity over time. And we approach this question using a particular type of pathology that is amblyopia, that is lazy eye. Probably, 5% of you will have a lazy eye, or at least if you have not been cured during development. So what we did, to make permanent, or try to make more long-lasting this effect, what we did, a counterintuitive therapy, we patched the amblyopic eye, we put the subject on a bicycle, and watching a movie. So we combine physical exercise with deprivation. We repeated the training just 6 times, every time for 2 hours. So overall, 12 hours of training. And this is what are the results. The visual acuity of the amblyopic eye dramatically decreased, improved during the training, nearly by a factor 2 line. You said, well, 2 line, but for these people 2 line meant a lot. And what is important, the association with the physical exercise really stabilized this homeostatic plasticity, so the effect was present after 1 month, 3 months, and after 12 months, a year. Importantly, some of these patients, not all, also some were really stereo blind, and they recovered some stereoscopic vision. So just in conclusion, and I am in time, I hope, visual cortex retain a high degree of plasticity in adults. Homeostatic plasticity induced by short-term monocular deprivation is associated with change in cortical excitability, a decrease of GABA, a boost of neural gain, phenomena that we do see during development in animal models. Physical exercise can be a physiological means to stabilize and to promote plasticity, and perhaps going on along this way, we can find also other strategies to endorse plasticity in other illness to promote self-repair. And I would like to conclude, remember I was Lamberto Lab many, many years ago in the 80s, and thank all the people in our lab in Pisa, the Pisa Visual Lab. Thank you.
Thank you, Concetta, very nice talk. And for being on time.